This is going to be the final video that contains content for Lecture 7. And in order for us to finish out the process of cellular respiration, we really need to understand where we stand so far. Following glycolysis in the citric acid cycle, we have completely broken down a molecule of glucose. We took that 6-carbon glucose and turned it into 6 1-carbon carbon dioxides. So all the carbons have been broken down. And all the electrons that were contained within that highly reduced molecule of glucose have been transferred into molecules of NADH, FADH2, and some of their energy has been utilized to create some ATP by substrate level phosphorylation, just not very much. Now these NADH and FADH2, they're not something we've talked about really yet, but these electron carrier molecules are carrying with them all that chemical potential energy from that big high free energy molecule of glucose. And they're who we're going to be paying attention to now, because they're going to donate the electrons they got from glucose to the electron transport chain. They're going to be this final part of cellular respiration, which you know as oxidative phosphorylation. Now, I want to recall for you that you know the structure of a mitochondria, but it's that structure which is going to be highly important for us to understand its function. Mitochondria are composed of 2 membranes, an outer membrane and an inner membrane. So enzymes like phosphofructokinase or triose phosphate dehydrogenase that are part of glycolysis are outside of that outer membrane. There's going to be a space between the two, which is known as an inter, with a T, intermembrane space. And this is going to be where all the protons are found in this complex. And then the inner membrane contains ATP synthase and other enzyme complexes which participate in this final step of cellular respiration. And it's what allows this inner membrane to become functional. These are the actual cellular respiration functional ATP making membranes. So the proteins that are in the membrane are giving the membrane its function. But this inner membrane is going to be folded in and around itself, creating these cristae folds. This creates a high amount of surface area, which allows us to embed more of these protein complexes, which makes these membranes even more efficient. So the process that we're going to be following, oxidative phosphorylation, actually happens in 2 steps. Step 1 is the electron transport chain. This is where these electron carrier molecules donate their electrons to this process. And it's actually going to go through the process of pumping protons to create and harness the energy from the electrons. Chemiosmosis is going to finally utilize that electron to create ATP. So the electron transport chain creates no ATP directly, but it transfers the energy from the electrons into a form that can be utilized by chemiosmosis to create ATP. In step 1, the electron transport chain, are electron carrier molecules, NADH, FADH2, which are carrying electrons from glucose's breakdown in our first stages of cellular respiration. These are going to actually pass the electrons to a molecule within the electron transport chain. Now, the purpose of that molecule is to accept the electrons and then utilize the energy from those electrons to pump the proton. So these electron transport molecules are going to be responsible for pumping protons into the intermembrane space. We've looked at proton pumps before. These proton pumps are transmembrane proteins that do active transport. In this case, though, our energy isn't coming from ATP, it's coming from the electrons, which are being donated by the electron carrier molecules. This is also going to be a pumping process which moves an ion across the membrane. So not only do we have differences in gradient, but differences in charge across this inner membrane of the mitochondria. And this is known as an electrochemical gradient, in this case formed by the pumping of protons. This is a very special and unique electrochemical gradient in the cell. And so, as we often do, when it's a very special, highly important form, we give it a special name. So this electrochemical gradient formed by the pumping of protons is named the proton motive force, because it's emphasizing the capacity of this electrochemical gradient to be able to do work. Now, the electrons are donated by NADH and FADH2 into the complexes. So here's complex 1, where NADH donates its electrons, and complex 2, where FADH2 donates its electrons. And these electrons are going to be systematically passed from one complex to the next complex, to the next complex, and to the next complex. Each time that these electrons are donated to a new complex, they're going to drop in free energy because some of that energy is being utilized to pump a proton. So it's important to recognize that there's work being done, the pumping of protons by these complexes, and that work is utilizing the free energy. Now, by the time our electrons get all the way down to this end complex, they are very low in free energy. It's a good thing that oxygen is highly electronegative, which means it has a high affinity for electrons. And because this oxygen wants electrons so desperately, it will accept even this very low free energy electrons and form water. This is the reason why aerobic cellular respiration is more efficient than anaerobic. Aerobic cellular respiration uses oxygen, a highly electronegative atom, and so it wants these electrons, and so this process is fast and efficient. Anaerobic cellular respiration is going to be using something other than oxygen, still have an electron transport chain, still do everything prior to and leading up to this, but we're going to use something other than oxygen here. And since that's not going to be as electronegative, it's not going to be as efficient. Oxidative phosphorylation comes in two parts. So remember that so far we've taken electrons from our electron carrier molecules, we've passed them through these electron transport complexes, pumped protons as we went, utilizing the energy in those electrons, and now that they're lower in free energy, they can be donated away to a molecule of oxygen, reducing it into a molecule of water. This ultimately creates no ATP, but it does result in the production of an electrochemical gradient known as the proton motive force. Recall that the proton motive force has the capability of doing work. Well, the work that it's going to do is when those protons flow through an enzyme known as ATP synthase. You might recall that we've talked about this when we did our membranes lecture, and we looked at transport across the membrane. This is an example of co-transport. This ATP synthase, as the protons move back through, we're going to turn a rotor in ATP synthase, which actually catalyzes the reaction by which we generate a molecule of ATP by combining an inorganic phosphate with ADP. This is different from substrate level phosphorylation, where an enzyme steals a phosphate from another molecule. Here, we're attaching a phosphate that's just an inorganic phosphate floating around the cell to a molecule of ADP to create ATP. And all of this depends on the power of a lot of oxygen in step 1, the electron transport chain, to accept electrons, because that drives the electron transport chain to continually move forward. If there was no oxygen, the electron transport chain wouldn't be able to give away electrons at the end, and so this entire process would stop. That's why we call this oxidative phosphorylation, because you need to have oxygen in order for this process to keep going. In order for us to calculate the amount of ATP produced per molecule of glucose, we need to be able to think about how much ATP is made per electron carrier. In this class, we're going to be using estimates. 2.5 ATP is produced per NADH, 1.5 per FADH2. Since we make 10 NADH molecules, 2 from glycolysis, 2 from the oxidation of pyruvate, 1 per pyruvate, and then 3 per turn of the citric acid cycle, and there are 2 turns, we have 10 NADH total. We also make 2 FADH2s, 1 for each turn of the citric acid cycle. And so if you have 2.5 ATP for each of 10 NADH, 1.5 ATP for 2 FADH2, that gives us a net of 28 ATP. In glycolysis, we also had a net of 2 ATP produced, and in the citric acid cycle, we had a net of 2 ATP produced. So we have 4 ATP, which is made in addition by substrate level phosphorylation. So 28 ATP by oxidative phosphorylation is clearly far more, and that's the majority of ATP made by cellular respiration. But don't forget, we still made 2 by substrate level phosphorylation. There are 3 major reasons why the number of ATP that you're being used in this calculation isn't the exact number, it's an estimate. The first is that this process is not directly coupled. There's a reason why you use electron carrier molecules to bring these electrons, generating an electrochemical gradient, and then utilizing that electrochemical gradient. There's no one-to-one transfer. There's no electron that's ever actually coming into contact with the ATP being produced by oxidative phosphorylation. And so you're not going to get a specific number. You don't get 1 ATP per electron. It just doesn't work that way. So that's why it's not a whole number. The ATP yield also varies depending on whether electrons are passed from NAD plus or FAD. And if this is the case, then you have slightly different numbers for NADH and FADH2, like we saw on the previous slide. Lastly, the proton motive force is sometimes used to do other work as well. Although ATP synthase is the only enzyme we're talking about that utilizes this process, the cell can also utilize this potential energy to do other work. And so sometimes, even if you pump 3 protons, you might not actually get 3 ATP out. Now, I want you to remember the flow of energy here to help you put all of this into a context. During cellular respiration, glucose is going to be broken down, and those electrons are systematically harvested by enzymes called dehydrogenases. They reduce electron carrier molecules by taking the electrons from glucose and its downstream products. Those electron carrier molecules bring the electrons to the electron transport chain. And it's the energy, the free energy from those electron-containing molecules, which is used to pump protons to make a proton motive force. And it's the potential energy of that proton motive force, which turns ATP synthase's rotor, and finally makes ATP at the end. So it's quite a step to go from glucose to electron carrier to electron transport chain to proton motive force to ATP. And only about 34% of the energy that's within a glucose molecule ever actually becomes ATP. Some of the energy gets lost as entropy, and that's why your body stays warm all the time, because some of that energy sort of gets dissipated into surroundings. Some gets spent on other processes in the cell and gets utilized for other things. There is a lot of vocab for you to know. Here is a review of vocab from 7.1 and 7.2, and here's some vocab from 7.3 and 7.4, as well as some key topics that you might want to consider and questions that you might want to ask yourselves. And now that you're done with this video, I want you to watch the BioFlix tutorial, which is next, and any other review materials that you're interested in, because video 7.5 doesn't have any new content. but it will ask you some questions that I think can help you test yourself going forward to see where you stand and how well you understand this material before the exam.